Yeah, so I think, I bring a slightly different perspective, right, to this webinar. I'll be more focusing, on proteomics. But as we all know, right, all the omics, all layers are very closely connected. What I would like to do maybe in the next 20 minutes, right, is to give an idea what we're doing at Biognosys in proteomics, and also, how we're looking forward to work together with our colleagues at biocrates in the future. Perfect. So a very short introduction to Biognosys as a company. So as already mentioned in the introduction, so we are a Swiss company located in Zurich, Switzerland. And we were founded around 17 years ago as a spin-off from Rudi Eberso's lab at ETH Zurich. I think many was a pioneer in the field of mass spectrometry-based proteomics. So since then, we've dedicated a lot of our efforts in developing technologies and platforms, both on the software side, but also on the wet lab side, to push mass spectrometry proteomics beyond its limits. And so far, we've been offering our products both as software, but also as a contract research organization. And since 2024, we also operate a lab in Newton, in Massachusetts, in the US. And since a bit more than 2 years, we are a part of the Bruker family as our colleagues at biocrates. What I would like to start with is really driven, let's say, an evolution driven by genomics. So as many of you have seen and also probably have experienced, the most dramatic change in genome sequencing is really the reduction of cost, while the quality and the depth of the sequencing can be kept. And this is really recapitulated in the plot on the left side. And the challenge we face here is that genome, as many of you know, is static. While if we move into transcriptome or further into proteome or even into the metabolome, things are getting more and more complicated and more dynamic. So you've heard, I think, in the talks today already that the metabolome is extremely dynamic, and just sampling time plays a key role, while proteome is less dynamic but still way more dynamic and diverse than genome and transcriptome, which means that we would require really high throughput, sensitive, and cost-effective method to be able to measure it, because taking one snapshot is in many cases not enough, so we would need to do it many times. And this is what shows the development of mass spectrometry proteomics in the last decade, or a bit more than a decade already. So as you can see, the field was really still at its infancy, right? Just 15 years ago in 2010. So there, let's say the technology is only sensitive enough, to measure the yeast proteome, right? Which is of course much simpler than the human proteome. And you can imagine that back then, mapping the entire yeast proteome would make it to a cover story in Nature, which is done nowadays in maybe 5 to 10 minutes. But the speed, how the field evolves, is really impressive. So you can see here, basically just 5 to 10 years later, a human cell line, for example, HeLa or a HEX cell line, can already mapped really at record time. Where, for example, at 2019, a publication by our team at Biognosys showed that for the first time we can map more than 10,000 proteins in a human cell line in a single mass spec run. And just a few years later, this number has by far been surpassed by the advancement of instrumentation and data analysis method. And by 2026, we think that basically the proteome coverage can really be achieved at the level of RNA-seq coverage without any amplification method. And this is just talking about depth and sensitivity. What's even more important is how this depth and sensitivity can be achieved with a reduction of cost. So as you can imagine, 15 years ago, it was an effort of the entire laboratory of 20, 30 people to map the yeast proteome. And things have developed slowly. So you see here in SPD, so basically samples per day, meaning that, let's say around 2018, we can analyze around 8 samples per day per instrument, which leads to a rough cost of around $1,500 per sample, which is by far too expensive. You think about a clinical cohort or a screening approach. What we already can achieve today, this is just 4 or 5 years later, is that we can already meet the same performance with around 40 samples per day and lower the cost by more than threefold. So basically, what we continue to observe here, right, is that the throughput is also increasing more than linearly, and we're trying also to reduce the cost. So if you remember the genomic plot there, we're really at the end really aiming for also $100 proteome, which would of course enable more and more data being generated and contribute to the scientific questions. Now going back a bit to the biochemical and analytical part of mass spectrometry-based proteomics. So what happens is basically the samples, biological samples, are collected, from human, from preclinical animal models, or from cell lines, etc. So how samples are prepared is it's quite straightforward and has now been standardized and automated. So protein will be extracted, denatured, and digested, typically using a enzyme called trypsin. And this basically yields tryptic peptides, which can then be separated on a liquid chromatography system that is connected to a mass spectrometer. And following that, we've developed software to analyze raw data and further interpret the outcome of such a study to answer different type of biological questions. I just list a few of them right here. So biomarker discovery is certainly one of the focus that we're looking at, but also since we're doing mass spectrometry-based approaches, we also have access to so-called proteoforms. These includes post-translational modifications, mutations, isoforms, etc. And this really helps to, let's say, increase the diversity of question that we can answer. So at Biognosys, we've created 3 different platforms to address different type of question. I will not go in that much into the details, but basically what we try to do is to take advantage of the versatility of mass spectrometry-based proteomics and try to really address different questions that arise at different stages of drug discovery. As you can see here, really starting from early stage target discovery down to clinical trials, where we are monitoring, let's say, the expression level of a single protein target. And this gives you, another, let's say, granular example of what type of exact biological questions we are addressing along the drug discovery pipeline. And I think this is also the strength in general of mass spectrometry-based proteomics, that it can really be customized to the question that has been asked. What I would like to cover a bit in the next 10 minutes or so is to introduce you also to some of the new developments happening at Biognosys. As you can see, right, a lot of the advancement have been driven by the advancement in instrumentation. But what's also important is to develop and optimize a platform around this instrumentation so that we can really generate robust and reproducible data in the long run. So what we've been focused on lately are around plasma proteomics. So there we're introducing a P2 plasma enrichment method, which allows up to 9,000 proteins unbiasedly quantified in human plasma. And then we are also complementing our mass spectrometry-based method with targeted assays based on affinity capture agents. So in this case, we're using NMR Biosciences ELISA assays. And last but not least, also to increase the power of sensitive detection, we've internalized cis-kappa assay, which uses antibody against peptides to be able to really look at very, very low abundance targets with very high selectivity and specificity. I think what's really interesting and what probably would serve as a great transition also for the next session is using blood as a matrix for biomarker discovery, because it's, of course, the most accessible surrogate matrix, right, in any human studies or trials. The challenge, as you can see here, outlined nicely in the publications, is first that the blood proteome is very, very diverse. It's far just from intact proteins, but it contains a lot of leakage protein, including cleaved products and let's say a tissue or cell leakage protein, which makes these a very, very diverse pool that is hard to analyze. And the second point that has been already brought to the attention more than 20 years ago is the very broad dynamic range of proteins in our plasma. And the challenge is really that the top 22 proteins made up already 99% of the total protein mass, while some of the interesting proteins, as you can see here, for example, the tissue leakage protein in case of of cancer, or signaling proteins, or cytokine chemokines in case of autoimmune diseases, and many more others, things like phosphorylated tau, etc., in case of neurological disorders, are even much more lower abundant and really requires a sensitive technology to monitor. And this, brings us really to the motivation of developing a new method to be able to do this in a robust and high throughput way. So we developed this particle-based system called P2. So what happens is we can incubate human plasma with this P2 particle, which leads to so-called corona formation. And this corona formation enriches for low abundant protein, while the higher abundant protein are depleted in the same process. And basically what happens is we developed a semi-automated workflow, which allows capturing of these proteins and then digestion and cleanup, leading to peptide, which we can analyze then using unbiased DIA mass spectrometry. And once we get this data, we can integrate them, of course, with clinical outcome data and other metadata, right, to make a data interpretation. I'm showing you just one example here. So here we're analyzing around 63 clinical samples from a phase 2 trial in lung cancer. And what you can appreciate here is that from these 63 samples, we managed to quantify around 9,000 proteins in total, where around 6,000 of these proteins could be quantified without any missing values across this. So this has been quite impressive compared to what mass spectrometry-based method could achieve in the past for plasma. And what's also very important, right, is to see how many drug targets either in development or in clinical trial or approved, right, could be covered in these 9,000 protein, because it's also important, to be able to monitor the protein that people are following and are interested in. You can see a quite significant portion of these drug targets could be monitored. I'm just using KRAS as an example, which can be very robustly quantified in this case in NSCLC, as well as in mesothelioma samples. And just as an example, we took two classes of protein that are typically either drug targets or pharmacodynamic biomarker that people are measuring. So one example is kinases, right? So we're looking at around 445 kinases directly in plasma. and cytokine, chemokine, which in the past were not, were basically below the limit of detection for most mass spectrometry-based approach. In this case, could also be assessed. So in this case, we could measure around 65 cytokine or chemokines, just showing, for example, IL-18 as an example here. I think what's also very interesting, I will not really go much into the details, right, is that that this data typically is very rich, right? In this case, around 6,000 full profiles. So there's a lot that can be done with these type of data. So one example we've shown here is using a method that has been created for transcriptomics, right? The weighted gene correlation network analysis. So here, what one could figure out are different modules, right, that correlate to a certain clinical features. So in this case, we could identify pathways, right, that are either upregulated in mesothelioma or non-small cell lung cancer, and seeing that basically these are two different type of diseases driven really also by very distinct biological pathways. And I think what's also very interesting, also what we learned from our clinical collaborators, that have, of course, direct contact with the patient, is that each patient, is of course a person, where we really more look at the data, as a heat map. So what we could do here is we're trying to turn these 6,000 proteins into a representation of hallmarks and pathways, and trying to see, how can we interrogate this data and translate it, to a language that clinical experts would understand. So as you can see here, right? So this again is from the same trial, where we just took a responder, one person, that responded to a treatment, and in this case, categorized the plasma proteome into different pathways that play a key role in, for example, in this case, in NSCLC and immuno-oncology treatment response. So you can see some of these familiar names popping up, right? JAK-STAT pathway, etc. And this, instead of looking at a single protein, these pathway heat maps really, gives these clinical experts an idea. Okay, for example, in pre-dose, we have, very low activity in some of the pathways, and upon treatment, some of these pathways seem to be turned on. And this can be, correlated in this case to a response to treatment. And if we take a non-responder using exactly the same methodology, we can see that, okay, there's already differences at pathway level at baseline, and upon treatment, the response to treatment also at pathway level might be different for certain things, and might be the same. So this really, gives a more personal, let's say, omics readout for on the clinical level. Kind of, to close my part of the presentation, I think what we learned from looking at a lot of proteomics data is that in many of these complex scenarios, right, oncology certainly is one of the most complex scenarios, not to mention, neurology or other metabolic diseases, that typically looking at one layer gives you some idea, right, as I've shown on the last slide, some idea on the pathways, on the potential mechanism of resistance or responses, but it never gives gives you a very clear-cut answer to the question, right? So this is really what's motivated us, right, to to start really looking into multiple layers of biology by doing what people call multi-omics, right? So I think this for us, right, multi-omics is really question-driven, right? You've seen that we already are doing a method like the ELISA approach, right, which allows us to look at some cytokine and chemokine expression, which is important in immunology and autoimmune indications. And we're also convinced, right, that by adding metabolites and lipids quantification, for example, using the biocrates Quantum 1000 kit, right, that you've seen also in previous presentations, that it also helps us to get another layers of information that may help, that may support and to help us to address some of these complex biological questions much better. And I think at the end, right, it should always, be a question driven approach, and we use really different omics to be able to integrate the data, but also to be able to get to a let's say more profound understanding of the problem. Yeah. So with this, I think just going to the take-home messages. I hope I could give you really a sneak peek, right, on mass spectrometry-based proteomics. And I think with the increasing throughput and decreasing cost, it has really the potential to become a routine tool, right, in drug development. And one of the key advantages is that it's extremely versatile, right, and can be used to address really different type of research and biological questions. And then the last take-home message is really that most of the biological question really we try to answer, especially if human samples and human diseases are concerned, are extremely complex, right? And typically we will require really data from a multi-layer omics approach to be able to get a much better understanding of the question. So with that, I would like to thank you for your attention.